Hello, my name is Ryan Lefebvre, and today I will be presenting the article titled Replication Errors Made During Oogenesis Lead to Detectable De Novo mtDNA Mutations in zebrafish oocytes with a low mtDNA copy number, written by these authors. I chose this article because in our Mechanisms of Human Diseases class, we covered several disorders that involved mtDNA mutations, and I found the topic to be rather interesting. What is mtDNA? mtDNA is DNA found in the mitochondria of the cell, but it has some very unique characteristics. It's replicated separately from nuclear DNA, and it can only be inherited from the mother. Additionally, it has a very high degree of variability, which is a result of several factors, but the most common of which is limited recombination. This means that there are fewer chances for recombination-mediated repair events to take place, which means that when mutations do occur, there's less of a chance for them to be fixed. Additionally, the mtDNA's proximity to oxidative phosphorylation and the reactive oxygen species that produces can increase the possibility for mutations to occur. mtDNA requires a high mutation copy number, or number of mutant mtDNAs in the cell to create pathogenicity. There have been more than 150 pathogenic mutations in mtDNA identified. However, there's a problem. In most patients who have mtDNA mutation disorders, the mutation can also be found in the mother. But in 25% of these patients, that is not the case. So, where is this mutation coming from? Well, it's most likely occurring to a new mutation, or de novo mutation, that happens in germline development. In germline development, you have a primordial germ cell, or PGC, that contains a certain number of mitochondria, some of which may be mutated. When this PGC then divides into primary oocytes, the mitochondria are randomly segregated into the new oocytes, which means that one oocyte may have a higher concentration of mutated mtDNA than the PGC. This means that further down the line, offspring could express a mtDNA disorder not seen in the parents. This is what's known as the bottleneck effect. The hypothesis for this paper states that with a constant mutation rate, the bottleneck effect could cause a low mtDNA copy number de novo mutation to result in pathogenicity. Previously, this hypothesis could not be studied due to limitations in the sequencing technology. However, recently improved sequencing techniques allowed the researchers to move ahead and assess this hypothesis. To start, they needed oocytes, and the researchers decided to use zebrafish oocytes. Zebrafish is a model organism used in many different branches of science, and its oocytes have mtDNA that is very similar to humans. They collected 103 oocytes from zebrafish and lysed those oocytes so they could extract the mtDNA. Then they put this mtDNA into PCR after cleaving it into 3 segments. The PCR reaction involved 3 sets of forward and reverse primers, one for each segment of the DNA, and it went through 40 PCR cycles. The first cycle, denaturation, went for 10 seconds at 98 degrees Celsius and split the double-stranded DNA into single-stranded DNA. The second step, which was 20 seconds at 58 degrees Celsius, was annealing. This allowed the primers to bind to the appropriate single-strand DNA. Finally, 8 minutes at 72 degrees Celsius allowed for extension, during which the polymerase bound to the primers and extended it along the template strand until new DNA was formed. The reason why this extension step is so long is that the segments of mtDNA used were quite large at over 5,000 bases. After they completed their PCR, the researchers checked their work with gel electrophoresis. This allowed them both to ensure that they had the appropriate mtDNA sequences and to check for any large deletions or insertions. Then they used next-generation sequencing, specifically Illumina sequencing. This is the new technology that allowed them to attack this hypothesis that they didn't have before. The first part of next generation sequencing is cleaving the sample into smaller sections. We saw that happen in the PCR section. Then the PCR product is flooded with fluorescently labeled nucleotides that also have termination sequences added to them. These nucleotides are then added to the template strand one at a time due to their termination sequence and analyzed for what color fluorescent tag they have. Each color is associated with a specific nucleotide, A, T, G, or C. This means that as they go along, they both determine the entire sequence and create a new strand of DNA until they have the entire mtDNA sequence. The benefits of the new generation sequencing are that it's cheaper, quicker, more effective, and you can use a smaller sample. After they had their results from the sequencing, they added some filters to make sure they were only looking at relevant de novo mutations. First, they had a threshold of coverage for mtDNA of greater than 1,700. This can be seen here in the blue line on the graph. The red line is the median number of mutations for each nucleotide position. So if more mutations are at a specific nucleotide location, then you will see a spike at that nucleotide location. Then they determined the heteroplasmy of each oocyte's mtDNA. That is the ratio of mutant mtDNA to wild-type DNA, and compared that to the maternal heteroplasmy. If they were more than 1% similar, then they were excluded from the study because they could not guarantee that those mutations were new in the oocytes and not previously found in the maternal tissue. Finally, variants that had multiple mutations within 1 to 2 nucleotides bases of each other were eliminated from the study. So what did they find? Well, from the agarose gel electrophoresis, they were able to see that there were no major insertions or deletions in the mtDNA sequences. Then they saw that there were 2,624 individual nucleotide-based substitutions. However, as they applied their filters, that number got smaller until they ended up with 38 de novo single-base substitutions. And those 38 mutations came from only 19 of the original 103 oocytes. They also calculated an average heteroplasmy for those mutations of 2.7%. Of those 38 mutations, 22 were non-synonymous, meaning that they don't code for the same amino acids as the wild-type DNA. This makes them more likely to be pathogenic as they could interrupt a protein's function. Additionally, there was no difference in mutation rate between codon positions found. So the A, P, and E positions of each codon, there's no preference shown for any one of those positions. Finally, they calculated the average mutation rate to be 4.3 times 10 to the negative 6th mutations per nucleotide location. What's interesting is that polymerase gamma, which transcribes mtDNA in the cell, has an error frequency between 2 times 10 to the negative 6th and 10 times 10 to the negative 6 mutations per nucleotide. This means that the average mutation rate fell right within the error frequency for the polymerase that codes for that transcribes these sequences. This graph shows the mutations based on the gene type. While it may look like the D loop is a significantly higher number of mutations, these results were not statistically significant, so they cannot say that D loop is more likely to have mutations than the tRNA, rRNA, or protein coding regions of the gene. This graph shows the mutations in individual protein coding genes. What's important about these is that most of these genes code for proteins involved in oxidative phosphorylation. So if you have a mutation in here that interrupts that protein's function, you also hinder the cell's ability to produce energy, leading to pathogenicity. From this, they were able to conclude that replication errors occur continually in all oocytes, but replication errors that occur during oogenesis can specifically create de novo mutations. And if these mutations occur at the end of the bottleneck effect, then there is a higher heteroplasmy level and increased risk for pathogenicity. They also determined that reactive oxygen species are less likely to contribute to mutations as the mutation frequency was found to be within the error frequency of the polymerase. Finally, location and heteroplasmy determine the significance of the mutation and whether or not it will be likely to cause pathogenicity. In the future, they look to be able to analyze all oocytes more in depth, as the current technologies still limit them to only analyzing mutations with a high heteroplasmy level. Additionally, the researchers determined that they would like to assess the risk in human oocytes, as there are fewer mitochondria, so the risk of pathogenicity is higher. Are there any questions?